Hello, good afternoon, everyone. Thank you for waiting for me to come to class. So we have to finish lecture. Actually, I don't think we can finish lecture 10 today, lecture 9 today, but we'll go through as much as we can, and whatever we don't finish, we will wrap it up next week. Previously, we were talking about recombinant DNA, where a piece of DNA consists of genetic material from different species. Okay, and we were trying to apply this recombinant DNA technology in medicine. Right? Specifically, we're trying to turn bacteria into insulin factories. Right? So to do that, we have to, number one, locate the gene of interest, and we could do that through the online genome browser. Okay? Thanks to the Human Genome Project, we can easily access these genetic information from the internet now. So we've located the gene of interest. Number two, we have to design primers, which are kind of like sticky tags that you put in the DNA and tell the PCR which section you would like to copy. And so we've made millions of copies of the gene of interest. And the next step would be to take those genes and put it into the plasmid. So to quickly recall, a plasmid is basically a circular DNA. And in the plasmid, there are several key features that I want to introduce to you. And I will just copy the picture from the PowerPoint slide. So this is a simplified plasmid. They are much more complicated in real life, but it doesn't really matter. That's the name of the plasmid. And over here, we have these restriction enzyme sites. Okay, so maybe I'll highlight them instead. So these are restriction enzyme sites. Okay, recognition site or restriction enzymes. Okay, we spent a little bit of time towards the end last time to explain how restriction enzymes work. They're like little scissors that would cut DNA at specific locations. Okay, these locations are typically complementary palindromic sequence. Okay, they look the same one way um, and backwards, forward and backwards. Okay, so in this plasmid, you can cut it open with PST1 at this location. You can cut it with equal R1 here, and so on and so forth. Right? If you see a recognition site on the plasmid, it means you can cut it with that enzyme. In addition, we have these uh, AMPR and TETR. These are antibiotic resistant genes. Okay, they are used as selection markers. Okay, so remember, I told you these are plasmid. They usually give the bacteria some kind of special properties. Maybe they give them an extra thick wall. Maybe they give them the ability to cling onto each other better, or maybe it gives it the ability to become resistant to a particular antibiotics. And so within this plasmid that we're going to be feeding the bacteria, there are two genes for resistance. Okay, so we have AMPR, which is ampicillin resistance, and we have TETR, which is another antibiotic. It's called tetracycline. Now, I will explain this a little bit more later on, but basically, if I if I give the plasmid to the bacteria in a reaction tube, how do I know whether the bacteria have accepted the plasmid or not? Like there's no way for me to tell just by looking at the tube, right? Not all of the bacteria will accept the plasmid. Only a small portion actually will accept the plasmid. But how do you tell the ones that have and the ones who haven't? Okay? So that's what the selection marker is for. If the bacteria accept the plasmid, if they take up the plasmid, they will now be able to grow in the presence of ampicillin or tetracycline. Or the ones that haven't take up the plasmid, they would be killed by the antibiotics. Right? So by putting these resistant genes into the plasmid, you would give the bacteria that has accepted the plasmid protection against these antibiotics. Okay? So that's like the short version. I will explain more later. So if you don't 100% get it right now, don't worry. We'll get to it. All right. So let's see what's going on here. Uh, this ORI, that's just the promoter, okay? Beginning of where the transcription is gonna happen. All right. So we're gonna have those tubes that I told you about, little tubes right here, and in the tube, that's where you will find the plasmid. Okay, you just buy them online, right, from biotech companies. And over here, we are going to have uh, the gene of interest. And you're going to have millions of copies of this gene because you've cloned it from PCR. You've cloned it from PCR. So let's take a closer look at the plasmid. I'm going to draw an even simpler plasmid here. We will say that there is a um, recognition site here for some restriction enzyme. And sorry, just give me a second. Let me try this one more time. This is a really difficult note to draw. And I'm going to erase a little bit of that. And I'll say, let's say this is TTTAAA, which means backwards is also TTTAAA. So this whole thing is DNA, right? But I specifically want to write out the sequence here. And then elsewhere on the plasmid, I'm gonna put a little thing here. Let's say this is the AMPR, ampicillin resistant gene. So this is the plasmid. Now our gene is gonna be, like this. And again, all these is like ACTGs, okay? But let's just say for the purpose of illustration, at the ends of the gene, at the flanks of the gene on either side, let's say you have TTT, AAA, and then same thing over here. Okay, so what you can see here is, again, this is the gene of interest. And what you can see here is the gene is flanked by the same recognition site as the one found in the plasmid. You see, it's the same one, right? Look, same one. Same. And if you're actually doing this in real life, you have to make sure that the sequence does not appear within the gene. And you only want it to be at the ends, flanking it. So what we're gonna do next is we're gonna, in separate reaction tubes, these Eppendorf tubes, that's what they're called. Um, what you want to do is you want to add a restriction enzyme. Okay? I'm just gonna call it restriction enzyme X. Doesn't matter. I just make it up. Okay, restriction enzyme X. And let's just say restriction enzyme X will cut this particular halidrome. And so what will it look like? Okay, give me a moment first. I have to draw this out carefully. Okay, so we will still have the ampicillin gene right here, ampicillin resistant gene. But now the enzyme is gonna cut open, it's gonna cut open the plasmid. It's gonna create these. Does anybody remember what they're called? I told you last class. I don't know if you remember. Okay, so it's basically gonna do a cut like that. Right, and then we have these, what do we call that? Anybody? That's quite all right. Sticky ends. Okay, they're called sticky ends. Why are they called sticky ends? Because they can stick back together. The TT here, TTAA here, is gonna be complementary to the AATT. So they could just, come back together because of the hydrogen bond, right? There you go. And in a separate tube, we will also be treating the gene of interest with the same restriction enzyme. Right? And then what's going to happen is it will not cut within the gene because we have to make sure that the restriction site does not show up, but it will chop off the ends, and then it will look like this. All right, and then the other half is just gonna fall off, right? This is just gonna be like useless. And same thing over here, we're gonna have the other half, which is just gonna fall off. It's not gonna do anything. So you have sticky N here, and you have sticky N here. Right? And the important point is right here. Let me write it in pink for you. Both plasmid and gene of interest have complementary sticky ends. After being treated with um, the same. restriction enzyme. Right? And what does complementary sticky end means? Complementary sticky end means like this piece right here can attach to this piece. And then this piece right here can attach to this piece. So if I mix the two together, then I can actually put the gene into the plasmid. Do you see that? Right? The yellow part can stick to the yellow part, the green part can stick to the green part. Now you might be thinking, hey, is it possible for the plasmid to just close back on itself? Yeah, that's possible. Okay. But we are hoping that would be a small percentage of the plasmid. Right, so over here, what we're gonna do is we're gonna mix them. Gonna put them together, mix in the same tube, and then we're gonna add ligase, which is the glue. Right? We want it to go in and then gets permanently sealed together. And hopefully in the end, you're gonna get a plasmid, and this plasmid will have the ampicillin resistant gene. And more importantly, it is going to have our gene of interest, which I guess I'll just color it blue, okay? Right there. Do you understand that? Please let me know if you have any questions so far. Okay, what's next? Well, we have the plasmid with the gene. Now we're gonna feed it to bacteria. Right, so like you would have again like another vial. Right, this one has like E. coli in it or something, and then you're gonna add the plasmid to it. Right, you're gonna add the plasmid to it. And there are different ways you can encourage the bacteria to accept the plasmid. You can apply a little bit of current to it. You can put them on ice. There are different ways, okay? And at the end, what's going to happen is you will have a bunch of these bacteria. Okay, bacteria everywhere. And some of them will have the plasmid. Oops. Let me zoom in a little bit. Some of them will have the plasmid. There's a plasmid here. There's a plasmid here. Okay, some of them will have plasmid. Some of them will not. So this one has been, if it accepted the plasmid, we call it transformed. Transformed. Okay, meaning accepted the plasmid. This one did not accept plasmid. And the problem is like you can't tell, like I said. They look exactly the same on the outside, right? Unless we're using that method where you turn them blue if they have accepted it. But that one is complicated to explain, so like I'm not gonna do that. I'm gonna use this ampicillin example instead. Okay, so all of them are sitting in the same tube, Some has accepted, some has not. How do you tell? How do you tell them apart? Okay. So what we're gonna do is we're gonna take some of these things and we're gonna grow them in a petri dish. And so this is a petri dish. It's a little bit 3D that I'm trying to draw. Okay. So here we have a control. Okay. So we have basic nutrients, and that's it. Okay. Just basic nutrients. And then you take these things, you take all of it, and then you put them onto this plate and you incubate it. You put it in 37 degrees and you let it grow. What do you expect to see? Do you expect them to grow or do you expect them to not grow? Well, these are all bacteria. Whether they have accepted the plasmid or not, they are bacteria. And if you give them nutrients, they will grow. In fact, you will have lots of growth. Lots of growth. Lots of growth. Sometimes they call it lawn growth. They grow so much, it's like a lawn. And this actually is our control. Wow, it seems so long ago that we learned about control, right? That's the control group. Right? This ensures bacteria did not die during transformation. Okay, like what if like when you shock them with the electric current, you end up like accidentally killing them, right? If that's the case, then they wouldn't be growing here. So this is the control group. This shows you they are all well and alive. But we still don't know which one accepted the plasmid and which one did not. And that's why we need to have a second play right here. Second play. Right, so we're gonna take you don't need to put a lot on them. You just put like a drop, spread it out, and then let them grow. So this one, we're gonna have again basic nutrients plus ampicillin. Okay. So what do you think? Do you expect a lot of growth, no growth, or some growth? What's gonna? Sorry, what was that?
Growth.
Yeah, some growth, right? Not a lot like here.
Yeah.
Yeah, there will be like some growth. Okay. Oops, what happened? Oh, I'm not sure what happened. Just give me a second. Oh, there we go. Let me see if I can. That was really, really, really weird. Okay, so some growth. Let me write down for you. Only transformed bacteria can grow in the presence of ampicillin. Okay? Because the plasmid, if you recall, has an ampicillin resistant gene. Ampicillin is an antibiotic. So the ones that are surviving are the ones that have accepted the plasmid. Those are the ones you want to mass produce. Those are the ones that contain the insulin gene. Those are the insulin factories. Okay? And everything else that didn't accept the plasmid would die here. Now, there are a couple of scenarios here. I'm going to walk you through it. Try to listen. If you try to grow it on the basic one, the control, and you see lots, but then nothing grows here, what does it mean? Lots of growth in control, no growth with the ampicillin. What does it mean?
Ampicillin kills those bacteria.
Yeah, it kills all of them, right? But why? Right? It probably means that none of them has accepted the plasmid, right? So that's a failed experiment. If you see no growth here and no growth here, what does it mean? It means all the bacteria are dead, right? Probably when you transform them, you did something wrong. You put too much current through them. You shock them all, they're all dead. So even with basic nutrients, they can't grow, right? So that's why we have to do it on two separate plates, right? To figure out what's wrong. Right? If I only do this one, if I only do the one on the right and nothing grows, then I don't know if it's because I killed them all accidentally or if it's because they didn't take up the plasmid, right? So that's why I need to have a control to compare it with. And that's it. That's how you turn bacteria into insulin factories. And they would basically produce insulin into the culture. You can extract them, purify it, and off you go. Are we good? I hope so. Any questions? What is the purpose of putting the gene into the plasmid? Okay, so the gene is the insulin gene, right? By putting the gene into the plasmid and feeding it into the bacteria will now transcribe and translate the gene. So if this is an insulin gene, it will get converted into the insulin protein, which can be harnessed to treat diabetes. Right? And it doesn't have to be insulin gene, it could be whatever gene that you need, whatever protein you need to mass produce. Okay? Every protein has a gene that codes for it. If I need a lot of, you know, blood clotting proteins, I can in theory put it into the bacteria and let it mass produce the protein for me. Okay. Any other questions? All right. I think this is like the hardest part of the lesson. Right? Like if you survive this, the rest should be relatively manageable. Okay, let's take a look at this. A gene is inserted into the plasmid, this plasmid, using E-core R1, which is a restriction enzyme. The plasmid is transformed into a tube containing E. coli. You cultured all the E. coli on a plate containing tetracycline. What result do you expect? So if you cut using E-core R1, that means the gene is going to go right here, right? That's where the gene is going to go. And if the plasmid is accepted by the bacteria, do you expect the bacteria to grow in the presence of tetracycline? All of it will die, all of it will grow, only small portion will grow. What do you think? Let's launch a poll and see. All right. All right. Well, definitely will not be this one. Okay, because if they accept the plasmid, they have the tetracycline resistant gene here, which would allows growth in tetracycline. Will all of it grow? Not necessarily, because even if you feed it to let's say you feed it to 100 bacteria, right? It's not going to be 100 that gets transformed, right? Only some of it will get transformed. So the correct answer is C. Only small portion will grow, just like the picture I showed you, right? Only some grow, right? Not like all of it. Let's try another question. This is a little bit more complicated. It looks like the same question, but it's not. This time, we are inserting the gene using small one. Small one is right here. So that means the gene's going to go right here, right in the middle of tetracycline. So inserting a gene here will destroy the tetracycline resistant gene. What does that mean? It means even if the bacteria is transformed, even if they accepted the plasmid, it will not protect them from tetracycline because the tetracycline resistant gene in the plasmid has been destroyed by the insertion of the gene. So all of it will die. Right? So whoever did this experiment, they made a mistake. They should not have inserted the gene using small one. They should have inserted it using some other stuff. Okay, I'll give you a moment to think that through. I have more examples for you. I just need to find them though. It's somewhere here. Just give me a moment. All right, let's try this one. Random plasmid. Okay, and I'm gonna ask you what happens. So there are ampicillin resistant gene here. This is another antibiotics. It's called chloramphenicol or just CAM for short. So if I insert using equal R1, what can I use as a selection marker? Okay, let me zoom in a little bit. Okay. If I insert the gene using equal R1, can I use CAM as a selection marker? In other words, like my agar play, the nutrients, can I do nutrients plus CAM? Is that going to distinguish the ones that has been transformed versus the ones that have not? Is it okay to use CAM? What do you think? Okay, so equal R1, you're going to put the gene right here. If I put the gene here, it's not disrupting CAM, is not disrupting the ampicillin resistant gene. So it doesn't matter. You can use CAM to select, you can use ampicillin to select, right? Both cases, it will kill off anything that does not have the plasmid, but any bacteria that has accepted the plasmid will be able to survive. What if I insert using small one? What do you think? Insert using small one. Can I select using CAM or not?
Yes.
That's right, you can. Because if you insert using small one, the gene goes right here, gene goes here. It will destroy the ampicillin resistant gene, but it will have no effect on the CAM resistant gene. So by growing them on CAM um, culture, it will only kill the non-transformed one. But I cannot use ampicillin as a marker. Ampicillin will kill absolutely everything. Doesn't matter if you have septoplasma or not, it's going to be dead because the gene got inserted right into the ampicillin. Okay, so similarly, insert with ALU1. You cannot select with CAM, but you can select with M. There is a file on Lumine, I believe. Let me just show you where it is. And you can just go down to, this is lecture 9. And you can see the study resources here. Is it study resources? Yeah, there you go. Transformation and selection marker document. So you can open that, and there are some examples and description. And then there are examples you can try on your own, and then the answers to come as well. Okay, so you can go through those, and hopefully it'll give you a better understanding of what's going on. All right. When is our quiz? Let me just double check. Quiz is gonna be the 7th, which is on the Monday. Okay, let me see. I'm not sure if I can finish everything today. If not, I'll push it to Wednesday. So that's that. Let's move on to the next concept. All right. So that's it for recombinant DNA. So another biotechnology we're gonna look at now is called DNA fingerprinting. DNA fingerprinting. Okay. So you might have heard of this technique being used to determine paternity, to use as a paternity test and to see who the father is, right? Or to determine, whether this is your child or not. You can use DNA fingerprinting. It's 99.9% accurate. First, let's talk about something called polymorphism. Polymorphism. Polymorphism is any differences in DNA sequence that can be detected between individuals. Uh, okay, someone just asked what day is the final. I don't have it on top of my head, okay, but it's gonna be like the last Wednesday, the next Wednesday, okay. So polymorphisms can occur in coding or non-coding regions. The coding regions are the exons, and the non-coding regions are the introns. Now, within the non-coding regions, there are these polymorphisms that exist as short tandem repeats, or STR, short tandem repeats. So they are short, a few nucleotides short, and they exist in tandem, one after the other. That's what tandem means. And they repeat themselves. So that's why they're called STR, short tandem repeats, like CAG, 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 CAG. This is called CAG4 because it's CAG repeated 4 times. Okay. So you have this STR in your genome. I have it. and they could both be CAG, but what's different would be the number of repeats, especially for people who are not related. Okay, so you might have yours repeated 4 times. My copy might be CAG repeated like 8 times. Right? So that's a type of polymorphism because we can actually use biotechnology to detect these differences. Okay, and then we can use it for DNA fingerprinting. Now, sometimes these short tandem repeats can cause problems. Okay? Huntington disease is a condition that occurs when a person kind of lose control of their muscles, and then they have uncontrolled movement of the hand or like sometimes of the head, and so on and so forth. Not to be confused with Parkinson's disease. Okay, anyway, it's a bad disease. That's the key point. Okay, you can look up more about it later on if you want. And there is a gene called the Huntington gene, HTT. If it mutated, then it's going to cause Huntington disease. In normal individuals, this gene has a short tandem repeats of CAG that repeats 10 times or 35 times. But for people with Huntington disease, the CAG is repeated 36 or more times. Okay, so what happens is when the DNA polymerase is trying to copy these genes during DNA replication, if they are short tandem repeats, then it's very easy to introduce errors. Okay? If you're supposed to copy it like 34 times, it's very easy to accidentally copy it an extra time. So now instead of repeating 34 times, it's going to be repeated 35 times. Okay. And if it happens again and again over time, then the short tandem repeats can get longer and longer and longer. And up to a point when it goes beyond 36, then it triggers the Huntington disease. All right. And for Huntington, it actually doesn't show up until later on in life, in late 40s and beyond. And by that time, people already usually have kids. And so if the parents have it, it's actually a dominant mutation. Okay. So if the parents have it, there's a 50% chance that the kids are going to have it as well. So, sometimes children find out that their parents have it, then they can actually do a genetic test to find out if they will have it too. But it's not an easy decision, right? There's a 50% chance that you will have it. It's a debilitating disease that have no cure. It comes down to whether you want to find out or not, right?
I have one question.
Sure.
If the repetition goes 37, I mean, the severity of disease is
Yeah.
increasing with the repetition.
So I don't think there is a correlation between the number of repeats and the severity of the outcome. But if you collect samples from different people, that's the range that's going to be causing diseases. Yeah, that's a great question. Okay, not all STRs are linked to disease. I just showed you the Huntington as a sample. But we can use STR. In other words, I don't want you to think of STR as like a bad thing or something like that. Okay, STR it's just something that exists in our body, and we can actually use them to identify people by using DNA fingerprinting. So let's say this person has um, AGAT, right? That's repeated 12 times. Someone else might have the same one, repeated 5 times, and so on and so forth, right? So each person is going to have like a unique set of STR length. So if we can separate them based on the size, then we can create a unique barcode that will be only for that particular person. Okay. But to create the barcode, we will need a method to separate DNA based on size and based on length. That technique is called gel electrophoresis. A gel electrophoresis. I'm just gonna show you a video of it. That is probably the best way to explain this to you. Just give me a moment. I will have to quickly look it up. Sorry, I'm usually more prepared than this, but I just came out of the meeting, so I didn't have time to. preload all these things. So just give me a moment. And of course, you have to watch the commercial. Hold on, let me wait for it to go away first. There we go. All right, so I don't need you to listen to the sound, okay? And we can just go ahead and I'll explain what's going on here. Okay, so first of all, you have a power supply that's going to be creating an electric current. All these tubes, those are the little tubes I was telling you about where all the reaction takes place. They're called the Eppendorf tubes. And in these tubes, you would have DNA samples, okay? There is going to be these pipettes that allow you to basically pick up solution and load it into the machine. So this is the reaction chamber. There is a negative terminal here and a positive terminal. It's important to remember the backbone of DNA is made up of sugar phosphate backbone. Okay, and the phosphate is negatively charged, right? PO4 3 negative. So if I apply a current that goes from negative to positive, it will actually push the DNA away because negative and negative repel. So in this chamber, what they're going to do is they are going to fill it up with reaction. Right here, let's see. Oh, they're going to put the gel in. So let me explain a little bit of what this gel is. This is called an agarose gel. It comes in a bottle of powder, and then you add the powder to some buffers, and then you microwave it, and then it melts. And then you pour it into a cask. It's kind of like making jello. And after it dries, it becomes a gel like this. So this gel, it looks like this. There is some slots at the top for you to put the DNA in. I'll show you in a second. If you zoom in onto the gel, you will see the gel is made up of these honeycomb structure. All right, and it's reminds me of this play structure that I told my kids to like an indoor playground, and there was one of these things. Okay, and for those of you who don't know what this is, basically like you would get in at the top and then you try to like weave your way through to the bottom. Right? So my kids challenged me. They're like, "Okay, I'll race you to the bottom." And I said, "Yeah, sure, no problem. Sounds easy enough." Right? But they were so quick to get to the bottom. By the time they get to the bottom, I was still like stuck somewhere here. What's the main reason? Well, it's because I'm so much bigger compared to they are, right? If you are small, you can weave through these little holes and bands quite easily compared to when you're big. And that's exactly the principle behind this particular biotech, okay? The gel electrophoresis. This is going to be the negative terminal. This is going to be positive terminal. And then the current is going to be going this way. Okay, so you will have the DNA here. So if the DNA is really short, it's going to be able to weave through these honeycomb structure quite easily, and they will end up further down in the gel. But if it's really big, then it's not going to be able to go really far. Okay, so perhaps I should tell you a little bit more. In this gel, this is a single lane, okay, and then you load your DNA sample here. I'll show you in the video. And then you apply the current, and the DNA sample will only be able to go down its length. It's not like the DNA sample is going to go like that. Okay, it's only restricted to its length. And if it's really short, it will travel further down the lane compared to the longer ones, just like this thing I'm trying to explain to you. Okay, so let's go back to the video and see what's going on here. Okay, we have the gel. You can see the slots up here. They are going to pour in the buffers to help conduct the electricity. And then you just fill it up all the way until the gel is covered. And there's a nice little bar there that tells you not to go over it. Okay, and then there are the samples. We have 5 samples to load here. And let's see what's going on here. That's the pipette. And that's the sample right there. That looks like very little, but don't be fooled by it. There could be like, thousands and thousands of copies of DNA in that stuff. Okay. Now you see this is blue. The blue is not the color of the DNA. The blue is actually a dye that they've added to the DNA sample. Okay, and I'll tell you what the dye does in a bit. Right? So they pick up the sample with a pipette. I've done this many times, so like I'm speaking from experience. Okay, so they are gonna insert the pipette and they will pump the sample into the slot. So you can see the sample actually sinks nicely into the little slots in the gel, right? And that's one of the functions of the dye. Okay, the dye increases the density of your sample, so when you squirt it into the gel, it sinks nicely into the bottom of the well. Without the dye and you insert the sample, it's just gonna pop right back up and you will lose the sample in the buffer. So that's why we need the dye. That's one of the reasons. Okay, so they load it, okay, and then they gonna have to change the tip and pick up another sample. And here we go. Just gonna load it one more time. So remember, each of these are separate DNA samples, and they will stay in their own lane. Okay, so here we go. All the samples are loaded. We are going to close the lid, right, with the negative terminal hooked up to the negative wire and the red to the red. Okay, they will insert it into the power supply, turn it on, and then you can select the appropriate voltage and also the current and then the time. And then you turn it on. And when you turn it on, sorry, you can see the bubbling. The bubbling tells you that the chamber is live and there is electric current going through this chamber. Right? It's very safe. You cannot touch the solution while this is happening because there's a lid. If you remove the lid, then the thing will stop right away. And this is a time elapsed. You can see the DNA sample is migrating towards the right side, which is where the positive side is, right? So the dye is we cannot see the DNA. Okay, what we are looking at right now is actually we can see the dye. Okay, we call it the dye front, and the dye it's actually two dyes with different size. So the smaller dye is going faster than the heavier dye, but the dye is not where the DNA is necessarily. Okay, we can tell the DNA is roughly around here, but it's not exactly here. What that means is you should stop the gel, you should cut the current when the dye is halfway through the gel. You don't want the dye to be all the way here when you turn off the machine. Because if the dye is all the way here, then that means some of the DNA sample might have lost, at the end of the gel already. So the dye helps you load the sample and also gives you a visual representation of where things are as the experiment is happening. So this is called gel electrophoresis. Okay, and at the end, we would take the gel out. We would see the blue line, but we cannot see the DNA. To visualize the DNA, you have to stain the gel with something called ethidium bromide. Okay, ethidium bromide is a molecule that will bind to DNA and it will glow, it will fluoresce when you shine UV light at it. Okay, so this is the gel, and each of these band represent a DNA band. Okay, so in this sample, right, we can see one band, two band. In this sample, also two bands. But this sample, we have one, two, three, four bands. So the multiple bands are just DNA of different size. So in these samples, we have like really short one, we have longer ones, and so on and so forth. Right? Now, this you can put this under like a UV camera and you can take a picture and print it out and you can then see the bands clearly. These are not the same gel. Okay, so this particular one we only have one DNA strand in the sample, and this is short. Okay, and this one is like longer than this one because it didn't go as far, right? Think of the play structure I was telling you about. The longer ones get stuck here, the bigger ones, right? Just like I got stuck. So the closer it is to the well, that's where the wells are up here. Right? The wells are up here. This is the negative, this is the positive. The closer the samples are to the wells, the bigger the DNA strand is going to be. But how big is it exactly? that's when you need to have what's called a ladder. This is a ladder. A ladder, it's a ladder or a ruler. Okay. So that's basically something you can buy online, and they are prefabricated DNA of certain size. So you run it along with your DNA sample, and then the package will tell you how long each of these lines are. Okay, and then you can say, oh, this one is roughly around 172, and this one is roughly around 268. So it gives you a way to like measure it basically. So that's gel electrophoresis. All right. I am going to show you DNA fingerprinting briefly, and then that will be it for today. I have to move the quiz. Okay? It's not. I won't be able to finish it today. Sorry, guys. Okay, so DNA fingerprinting. So let's say we have a person here. We'll call him. We'll call him Lyle, and we will look at three of his chromosomes. Yeah, so let's say this is chromosome 2, and then there is STR right here. And this one is ACC repeated 10 times. And then we're going to look at chromosome 7 and chromosome 14. On chromosome 7, there is AT repeated 5 times. And over here on chromosome 14, there is CCG repeated 15 times. Something like that. Okay, we have another person. This is Ava. And we will be looking at the exact same DNA for Ava. Same chromosome, I mean. So chromosome 2, chromosome 7, chromosome 14, and Ava would have the same STR. You have the same one as me. Again, it's not a question of whether you have it or not. It's a question of like how long it is, right? So for Ava, her ACC here might be repeated only 5 times. And for the AT here, it's also repeated 5 times, just like Lyle. Maybe they have the same ethnicity, so it's possible that they have the same STR. And over here, the CCG, it is going to be repeated, I don't know, 20 times or something. I'll give you a moment to catch up, and I'll tell you more. Now, we know exactly where these STRs are because we have the information, the data from the Human Genome Project. We can just look up where these STRs are. And so we can design primers that would flank the STRs, right? And I will have like blue primers here that would flank this one and so on and so forth. And what's going to happen is we can amplify, we can get a sample from from Ava and Lyle, and from the sample, we can perform PCR using these primers. So in the end, what's going to happen is we're gonna have one of those tubes, and in the tubes we will have thousands of copies of the red DNA, so to speak. It's not literally red. We will have thousands and thousands of copies of the blue one, and then we would have the green one as well. All right. So this is clone using PCR. Okay, same thing here, PCR. Then we're gonna run the sample on a gel. Okay, sometimes we just draw it like this for the slots. It's easier to draw this way. Don't get confused by it. All right, so there is my gel. This is the negative. This is the positive. Let's put Lyo sample here and let's put Ava sample here. Okay. So where do you expect the lines to be for Lyo? Well, let's take a look. This one is ACC, which is the three nucleotides repeated 10 times. 3 multiplied by 10 means this is 30 nucleotide long. This one is AT repeated 5 times. 5 times 2 is 10. This one is 3 repeated 15 times, which is 45 nucleotides long. So please tell me, do you expect the blue band to be closer to the top or closer to the bottom for Lyle's sample? Closer to the top or closer to the bottom? Let's do a poll. Oh, I already have answers coming in. Yes, it would be closer to the bottom because 10 is relatively short compared to 30 to 45, right? It would have no trouble weaving through all those honeycomb structures within the gel, right? Making through the obstacle course easily, and it's going to end up all the way down here. So that's going to be one band. And remember, you cannot see the DNA unless you stain them with the ethidium bromide, but I'm just drawing it out for you to visualize it. Okay, we're gonna have a 30 probably somewhere here, and then the 45 is even gonna be higher up. Now, you know what? Let's move them a little bit down here. Right. Do you understand why the bands are where they are? Okay, let's take a look at over here. Uh, for the ACC, this is multiplied by 5, so that's gonna be 15. This one is gonna be 10, and this one is gonna be 60. Right, so 60 is longer than any of these. It's going to be like really high up here. And then for the 15, it's further down at the bottom. And then for the 10, it's going to be closer to 15, but same level here. Right? So just by using 3 separate STR, we can distinguish Lyle and Ava in this example. But in real life, we're not going to be using just 3 STR. We're going to be using like a lots of these STRs throughout the entire genome. And so the person is going to have a very unique barcode, right? So someone else might have that. So this barcode here, that's as unique as your fingerprint, but on a DNA level. That's why it's called DNA fingerprinting. All right, let me show you the slide here. So let's say in the crime scene, right? There is a sample, and then you do the STR analysis, and it looks like this. Then you have 3 suspects, right? So who's at the crime scene? It's suspect number 2. This guy has the exact same banding, right? Each of these band represents um, a DNA STR, right? And no one's gonna have the same thing as you unless you have an identical twin. Okay, we'll skip these things for now. Let's take a look at this question, and then we will end. Okay, so let me copy this over. All right, let's take a look here. Which of the following statement is consistent with the result? Right, so we have 4 individuals here, and this is a family of 4. Okay, I should tell you that. Family of 4. Uh, there are parents and kids here, and we want to figure out like Who is who? How do you even go about doing this question, right? So, well, let's see. Right? We know kids have to inherit stuff from the parents, and not the other way around, right? Okay. So whatever the kids have, the parents have to have it. Okay. At least one of the parents have to have it. So let's see. B is the child of A and C. B is the child of A and C. Let's see if it's possible. Right? So let's see. We're looking at B here, and we are assuming A and C are parents. So let's see. Okay, so B has this one. Well, did it get it from A or C? Uh, got it from C. No problem. Let's take a look at the next one. This one got it from C. And then the next one, that's where the problem is, right? There is a band here, but I do not see this band in A, and I do not see this band in C. That tells me A and C are not the parents of B, right? Because whatever B has must have come from one of the parents, right? And this didn't show up in A or C, which means that is not true. B is not the child of A and C, and the telltale sign is this one, and actually that one. Right? All the ones I've circled didn't show up in A or C. So that's why I know they're not the parents. Let's check the next statement. C is the child of A and B. C is the child of A and B. So let's see. This one came from here. Okay. This one came from A. This one came from B. This one came from A. Basically, you're checking to see if the band appears in either A or B. And if they all showed up in either A or B, then they are the parents. Right? This one, I like. I mean, it shows up both. Like, so I don't know where it came from, but doesn't matter. It doesn't affect our answer. So that's the right answer. Okay? C is the child of A and B, and that's how they do DNA testing. Right? If you want to find out like who the parents are, right? That's what they do. Okay? You can check the other ones to see why they're wrong. Okay? Let's try the next one. Okay. Which of the following statements is most likely true? Okay, so let's try another one here. D is the child of A and C. Why don't you guys figure it out? D is the child of A and C. I'll give you a moment. You tell me whether that's correct or not. So again, the name of the game here is to find a band that in D that doesn't show up in either A or C. If you find a band like that, then they are not the parents. Okay, so like this band right here, that is not in C and not in A. That's how I know D is not the child of A and C. One more time. If you can find a band that does not show up in two other people, those people are not the parents. Okay? So B is correct. B is correct. So if C is the child of A and B, parents, and D is the child of A and B, then what are these people? Well, they are siblings, aren't they? Siblings. C and D as siblings. All right. Okay, that's all. I mean, I don't have a lot more things to go through, just like nine more slides. Uh, but I don't want to rush through it. I feel like if I, mm, you know what? Uh, we have 10 minutes. Yeah, okay. You know what? Let's not rush through it. Okay, it would be unfair to you. So I'll move the test, move the quiz to Wednesday. Okay, on Monday we will finish the rest. We will also start the last lecture. Okay. So print the notes. Thank you. See you then.